Alright, so we've already talked about geological time, we've talked a little bit about fossil evidence, now we're getting into the point of early history, and we do have to have this as a background as well. Still looking at that amber image, because we're going to talk a little bit about how we get to the point of talking of that early history. So this is a lot of work just to go through all this information, so I'm not going to really go through and talk about I'll talk about each one rather than reading them. So there's a lot of different ideas about how the Earth formated. There's hypotheses about Earth's early history, and much of what we actually have about those hypotheses actually is based on a small amount of evidence. There's not one true story at this point. We do know that one thing we do know for sure is that it wasn't one event that just kind of went poof and there it was. There's a lot of things that happened, and we also know that while the Earth was young, looking at other planets as they formed and looking at other bodies, we were able to do a lot of research onto what happened to change even the Earth, because the Earth itself had to change to be able to be inhabitable by the life that's on it currently. There's a lot of gaps, and that's part of what we're looking at is some of those gaps. We've got a lot of scientific ideas about how things got here, and this is kind of what we're looking at here, is looking at some of those in-between parts where we talk about what do we know and what are we still trying to figure out. So things we do know is that at one point the Earth was completely inhabitable. We do know that there is a crust on the Earth. We do know that that crust became an important factor for us because it came to the surface on the top of the core of the planet, so on the outside edges on that crust became our land masses, and those land masses we know have moved. We know that they've shifted, we know that they continue to shift, we know that they're floating masses that actually allow for us to have the changes in the shape of our land masses that we currently have. So things to know. When we talk about the early atmosphere, what we have kind of come to determine is that it most likely had a lot of things that would make us die. Hydrogen cyanide, we would die. Carbon dioxide we still have. It is not great for us, but we definitely still have it in our air. It's actually one of the highest percentages. Carbon monoxide, that on the other hand is the stuff that we talk about that is poisonous, meaning that if you were to be by a car and it is releasing exhaust, that's going to be carbon monoxide, and carbon monoxide is poisonous to humans and other mammals. There are some organisms that can handle it, not a lot though. Nitrogen, we still have lots of nitrogen in our air right now, but usable nitrogen versus non-usable nitrogen. Hydrogen sulfide, again, not usable to us at all. Sulfide being that we're looking at it being acidic, and that would be dangerous to life on the planet. And the other part in our early atmosphere is water, that water vapor that's always kind of there. Okay, so we always are looking at those organic molecules. So thinking back to some of our organic molecules that we were looking for, the big one is amino acids. That becomes kind of our building block that allows us to be able to create just about anything else. So what ended up happening to be able to kind of figure out how we got to those amino acids or some of those very first organic molecules, we want to know how they actually existed, considering all the environmental conditions were poor for our current life. So in the 1950s, Stanley Miller and Harold Urey tried to answer that question. They went through and did some experiments to do that. This is their experimental setup that they used. And they were able to mimic Earth, and this is their kind of their mimic of Earth here, is looking at those atmospheric gases and the water vapor, knowing that it's uninhabitable at this point. And what they actually ended up doing is because the Earth, when it was young, was very violent. There was lots of storms, and there was a violent atmosphere, or violent weather, which is pretty common for young planets. So they actually did some spark stimulus that would actually simulate the lightning that existed. And what they actually found is that lightning and those chemical reactions that happen with that lightning and these gases over here, this mixture of those gases of the early atmosphere, was actually something that became an important factor. So after we had that lightning storms and you were actually able to see that chemical reaction, what we were actually able to do in that condensation chamber, where we actually collect then the liquid from the atmosphere again. The cool water actually cools the chamber, allows for those droplets to form, and what we found is that we had now had liquid containing amino acids and other organics. And this became huge. So this experiment has been done many, many times to kind of figure out if this really was something that was happening or if it was something that Urey and Miller had just kind of come up with. This is the setup of your experiment. It has been tested multiple times. It is something that we've been able to replicate and have been able to do at large scale and small scale. So how did life really get started? The real question is, how did we get to the point of having from pretty much nothing, where we just had organics, the beginning, to where we want to get to at the end, where we actually have that full organism with life? So it started with protenoids and microspheres. They're not really cells. They have organic material. We call them organic molecules, and they're in these little tiny bubbles. They're not cells. They don't have a nucleus. They don't have structure in which they're all working together to be able to do a job. They do, though, have selectively permeable membranes, meaning they can allow material to travel in and out. They can store and release energy, so they do have the ability to have something similar to a mitochondria, where they actually store and then release or make energy. So there's hypotheses that actually show that these structures, similar to these microspheres, might have actually become eventually our living cells. So one of the questions that comes up is always RNA and DNA. Which one came first? There's actually a couple different philosophies in this, kind of two big camps. One in which we say the RNA sequence actually helps make DNA, and the other one in which says that DNA had to be there first, otherwise we can't get to that RNA because we go through translation. Now, we know that we can probably go either direction at this point, but these are the two big ideas, and these two big ideas really have set up an interesting debate that they don't really know the answer to entirely yet. There's leaning towards the idea of RNA, but there's enough evidence to show that DNA might have been the first one. Okay, so let's talk about RNA and its origin of life. This is kind of one of those things that we're looking at. So we've got that stew, or that gunk that was created, or that in the bottom of that beaker. And we have simple organic molecules that are formed. They form RNA nucleotides. RNA can synthesize itself, which makes it part of the reason that we're able to argue for RNA being the first one. It actually then allows for us to go through protein synthesis, being able to have RNA make proteins, have those proteins then create cell structures and catalyze chemical reactions, because remember that proteins do all of the above. And the other thing is that DNA can actually be formed from our RNA as well. So it's an interesting thing, but RNA is kind of the direction everybody's going. Okay, so the key to get to where we are today is to look at the idea of free oxygen, oxygen that's available for use by organisms. The first life forms had to survive without oxygen because, as you saw, there really wasn't any in the atmosphere. And what ended up happening is that we did have some photosynthetic bacteria that began to pump oxygen first into the oceans, which is why life started in the water rather than on land. And then eventually we're talking about that oxygen gas accumulating and being able to be released into the air and then accumulating in the air, which led organisms to be able to leave the water and be able to be on the land. So how did that happen? So really, realistically, what we're talking about is we have those little tiny organisms, microorganisms, in which we have metabolic pathways that allow for us to be able to survive. So when oxygen was actually introduced, organisms had to be able to adapt or not survive. So extinction did happen. That was one of the great mass extinctions that existed. Other life forms then evolved, changed, adapted, and became so that they actually used oxygen as part of their metabolic pathways, as part of their respiration, to be able to make energy. And then that allowed us to be able to have the changes that we've seen and get to the point where we are. We have the complicatedness of those organisms that live on land and in water right now. So this is looking at going beyond those protenoids and getting into what we call our eukaryotic cells. Remember, eukaryotic cells, as a review, are like our plant and animal cells. They have a nucleus, they're membrane bound, and they have organelles in them. Okay, so endosymbiotic theory basically says that we had some prokaryotes that were ancient, and some of them did different jobs. Some of them were aerobic, meaning they used oxygen. Sorry, guys. And the other ones were photosynthetic, meaning that they made their own food. So what we're seeing is that we had things that were, we had prokaryotes that were ancient that were actually anaerobic. And what ended up happening is they ended up engulfing or surrounding that aerobic so that we could actually end up making what it needs. Now the aerobic bacteria means it takes oxygen and it actually is going to use it to be a metabolic pathway to build and create structures. In this case, our structure in releasing energy. So we are creating our organelles. So this is going to become that mitochondria using oxygen. The other one is that chloroplast, photosynthetic, which allows for us to make our own food or be able to create structure of sugars. So this is the animal-like, this is the plant-like, and being able to have that within our system. This is endosymbiotic theory. So realistically what happened is really they started living near each other, found out that they could benefit from each other, and they created a symbiosis, one engulfing the other, and they both benefited from it. Okay, next part is looking at the idea of sexual reproduction and the fact that with prokaryotic cells they produce asexually, meaning that their daughter cells are exact copies, and then this actually makes it so that there's very little change in genetics. The only changes that you see in those genetics are mutations. Otherwise, they're all identical. And we've talked about the fact that variation is difficult. Not having variation is difficult. Sexual reproduction does all sorts of things. It shuffles genes, creates new combinations, increase probability for favorable combinations, and then we also talk about the fact that there's evolutionary change due to natural selection. Those adaptations occur because someone has a great trait that they want to use. Okay, that takes care of that. We have one last little bit, I believe.